So in the last section, we considered the first stage of biological development. That was the pre-embryonic stage, the first 16 days, where we considered cleavage, implantation, and embryogenesis. Now we'll move on to the embryonic stage. The embryonic stage begins when all three primary germ layers are present. Usually that's about 16 days after conception, and the conceptus has implanted on the uterine wall. Over the next 6 weeks, the placenta will form. It becomes the embryo's primary source of nutrition. The process of organogenesis will also begin, where the germ layers will differentiate into the organs and organ systems. At the end of 8 weeks, when all organs are present, the embryo becomes a fetus. Organs are still far from fully functional, but this does mark the transition from the embryonic to the fetal stage. The embryonic stage is a conversion of the flat embryonic disc into a somewhat cylindrical form. It begins to occur around week 4. The embryo will grow rapidly and fold around a membrane called the yolk sac. The embryo becomes a C shape with a head and tail almost touching around the yolk sac. The lateral margins of the disc will fold up and around the sides of the yolk sac to form the ventral surface of the embryo. In this figure, you can see both longitudinal sections and cross sections of this process as the embryo grows around the yolk sac and as the sides of the plate fold up to form the neural tube. As a result of this embryonic folding, the entire surface is going to be covered with ectoderm. That's the outside layer, so it all makes sense. This ectoderm will later produce the epidermis. At this point, the mesoderm will split into 2 layers. One adheres to the ectoderm, and the other adheres to the endoderm. The splitting of mesoderm into 2 layers will form the coelom. The coelom is the body cavity that's between the 2 layers of mesoderm. So essentially, the endoderm will become the inner gut tube and its associated structures, and the ectoderm will become the skin and its associated structures. And then there's a space in the middle called the coelom. And then, as you know, outside of the gut tube, but inside of the body wall, there's a space. This space is the coelom that we see in a developing fetus. The body cavity will be divided into the thoracic cavity and the peritoneal cavity by the diaphragm. By the end of week 5, the thoracic cavity further subdivides into pleural and pericardial cavities. We'll see the appearance of the neural tube. The neural tube will become the brain and the spinal cord. In addition, we'll see the appearance of somites. Somites are remnants of segmentation that we see in the mesoderm, and it will give rise to the vertebral column, the trunk muscles, and the dermis of the skin. The formation of organ from the At 8 weeks, all organs are present in a 3-centimeter long fetus. Again, they're not mature or functional yet, but they are present. The heart is beating and the muscles exhibit contraction. Let's take a quick look at the derivatives of each of the germ layers. The ectoderm will give rise to the epidermis, the nervous system, the lens, and the cornea, as well as the internal ear. The mesoderm gives rise to the skeleton, muscle, cartilage, blood, lymphoid tissue, gonads and ducts, as well as the kidneys and ureters. And the endoderm will give rise to the gut and respiratory epithelium and the glands, the bladder, and the urethra. So basically, the ectoderm gives rise to everything on the outside of the body. The mesoderm gives rise and the endoderm gives rise to all the tubes on the inside of the body, and the mesoderm gives rise to everything in between. By the end of 8 weeks, all organ systems are present. The individual is about 3 centimeters long, and it's now considered a fetus. The bones have begun to calcify, and the skeletal muscles exhibit spontaneous contractions. These are much too weak to be felt by the mother, but the baby's moving in there nonetheless. The heart has been beating since the 4th week and is now circulating blood successfully. The heart and liver are very large, and they form a prominent ventral bulge on the fetus. And the head is nearly half the total body length. Take a moment here to outline what things make an individual a fetus. What things make an individual an embryo? What are the big events that go on during each of these stages? Now let's consider some of the accessory organs. Several of them develop alongside the embryo. We know the placenta is necessary, and the umbilical cord connects the fetus to the placenta. And there are 4 other embryonic membranes: the amnion, the yolk sac, the allantois, and the chorion. The amnion is a transparent sac that develops from the epiblast. It grows completely to enclose the embryo, and it's penetrated only by the umbilical cord. It fills with amniotic fluid. You can see that amnion here. The yolk sac arises from the hypoblast cells opposite the amnion. It's a small sac that's suspended from the ventral side of the embryo. It contributes to formation of the GI tract, blood cells, and future egg or sperm cells. Kind of interesting to think that humans even have a yolk sac, but that's one of the characteristics of vertebrates, all 4 of these membranes. The 3rd membrane is the allantois. The allantois begins as an outpocketing of the yolk sac and forms the foundation for the umbilical cord. It'll become part of the urinary bladder as the fetus matures. Finally, the chorion. It's the outermost layer, and it encloses all the rest of the membranes, as well as the embryo. You can see the chorion here surrounding the outer edge of the uterus. The chorion has shaggy outgrowths that are called chorionic villi. These chorionic villi grow around the entire surface, and as pregnancy advances, the villi of the placental region become particularly prolific and grow and branch, while the rest of them seem to degenerate. And thus we have smooth chorion and then chorionic villi in the region of the placenta. The villus chorion is where the placenta attaches, and it forms the fetal portion of the placenta. Over the course of gestation, the conceptus is nourished in 3 different but overlapping ways. There's uterine milk, there's trophoblastic nutrition, and then placental nutrition. We have to have the prior 2 because the placenta is not in place when the embryo first implants. Uterine milk is a glycogen-rich secretion of the uterine tubes and endometrial glands. The conceptus absorbs this fluid as it travels down the tube and lies free in the uterine cavity before implantation even occurs. Trophoblastic nutrition is when the conceptus consumes the decidual cells of the endometrium. Progesterone from the corpus luteum stimulates these decidual cells to proliferate. They accumulate and store glycogen, proteins, and lipids, all the fuels that the conceptus could possibly need. As the conceptus burrows into the endometrium, the syncytiotrophoblasts digest them and supplies nutrients to the embryoblasts. Remember, the syncytiotrophoblasts are the finger-like projections of multinucleate tissue that are weaving their way down into the endometrium. Trophoblastic nutrition is the only mode of nutrition available for a week after implantation and remains the dominant source through the end of 8 weeks. Its role wanes as the placental nutrition increases. The beginning of the 9th week is the placental phase. This is where the placenta develops. It becomes the sole mode of nutrition by the 12th week until the time of birth. In placental nutrition, nutrients diffuse from the mother's blood through the placenta and into the fetal blood. You can see a timeline of which form of nutrition dominates during which phase of pregnancy. So now let's take a quick look at how the placenta develops, or placentation. The formation of the placenta occurs between 11 days and 12 weeks. And as I've already introduced, the chorion produces chorionic villi, little extensions of the syncytiotrophoblasts that burrow into the endometrium by digestion and growth of roots of syncytioblast tissue into the endometrial layer. These are the early chorionic villi. You can see them here. We'll see mesenchyme extending into the chorionic villi from the embryonic blood vessels. If we go back a few slides, we can see this. In this diagram, you can see that these roots are the early chorionic villi. As they penetrate the uterine blood vessels, they become surrounded by lacunae, or endometrial spaces that are filled with maternal blood. The lacunae will then merge and surround each of the villi. This high perfusion of blood will stimulate rapid growth of the chorionic villi, thus stimulating growth of the placenta in general. The umbilical cord contains 2 umbilical arteries and 1 umbilical vein. The circulation of the placenta on the fetal side is pumped by the fetal heart, and blood flows into the placenta by way of the umbilical arteries, and it flows out by way of the umbilical vein. The chorionic villi are filled with fetal blood, and they're surrounded entirely by maternal blood. So the blood streams do not mix whatsoever, but the barrier is only about 3.5 micrometers thick. That's half the diameter of a red blood cell itself. This short distance allows diffusion of materials between the fetal and maternal blood supply. As the placenta grows, the villi grow and branch, and their surface area increases. The membranes become thinner and more permeable, which increases the placental conductivity as the fetus grows. The materials diffuse from one side of the membrane when they're more concentrated to the side where they're less concentrated. So that means oxygens and nutrients will pass into the fetal blood, whereas fetal wastes will pass back into the maternal blood. The placenta is also permeable to nicotine, alcohol, and most other drugs that could be present in the maternal bloodstream. So although there's no mixing of the fetal and maternal blood, many of the substances that we consume can cross the placenta quite easily and have great effects on the developing fetus. This is a table that summarizes many of the functions of the placenta. It has nutritional roles, excretory roles, respiratory roles, endocrine roles, and immune roles. The fetus is the final stage of prenatal development. From the start of the 9th week until birth, the organs will mature to support life outside of the mother. The circulatory system shows the most conspicuous anatomical changes from the prenatal state, which is dependent on the placenta, to the independent neonatal or newborn state. The unique aspects of fetal circulation are the umbilical placental circuit and the presence of 3 circulatory shortcuts called shunts. The internal iliac arteries give rise to the umbilical arteries, which pass on either side of the bladder into the umbilical cord. The blood in these arteries is low in oxygen and high in carbon dioxide. You can see the umbilical arteries marked by 3 here. The umbilical artery blood discharges wastes into the placenta and then loads up with oxygen and nutrients and returns to the fetus through a single umbilical vein. Again, this is one of those cases where the arteries and the veins are sort of reversed in their normal means of carrying oxygenated blood. The umbilical vein is carrying more oxygenated blood, which has been picked up at the placenta, whereas the blood in the umbilical arteries is much less saturated by oxygen. The umbilical vein will carry some of the venous blood through the liver to nourish it. The immature liver is not capable of performing many of its postpartum functions, and most venous blood bypasses the liver by way of a shunt called the ductus venosus. which leads directly to the inferior vena cava here. Within the inferior vena cava, the placental blood mixes with the fetus's venous blood and flows into the right atrium of the heart. It bypasses the fetal lung because there's little need for blood to flow into the fetal lungs because they're not functional yet and will not be involved in oxygen exchange. Thus, most fetal blood bypasses the pulmonary circuit at this point. There's a little hole between the atria in the interatrial septum called the foramen ovale. Some blood goes directly from the right atrium through the foramen ovale and into the left atrium, and some blood is pumped from the right ventricle into the pulmonary trunk. Most of this blood is shunted directly into the aorta by way of a short passage called the ductus arteriosus. The lungs only receive a trickle of blood sufficient to meet their metabolic needs during their development, so just enough so that the tissue itself can grow. Blood will then leave the left ventricle and enter the general systemic circulation. Some of this blood will return to the placenta. Now, these fetal circulatory patterns change dramatically at birth when the neonate is cut off from the placenta and the lungs expand with air. We'll look at those adaptations shortly. Take a moment now, though, to diagram the fetal blood flow between the placenta, the lungs, the heart, the liver, and so forth. Much like we did the diagram of the systemic and pulmonary circuits. But make note of which direction blood flows. It flows directly from the right atrium through the foramen ovale into the left atrium. It bypasses the lungs. Most of the blood also bypasses the liver. So take a moment to sort this out for yourself. It'll be part of our diagramming assignment for this week, so you may as well do it right now.